All right, folks. Apologies for the lateness. It might be an issue going forward. So I'm going to propose that we start class at 8:15 instead of 8 o'clock. We'll just go straight until 10 to compensate, or close enough to that. The road is far worse than I thought it would be for this time of year. Anyway, so my apologies for that. I sent a message in the group. I don't know if y'all got that message or not. Did anybody see the message I sent?
Yes, sir.
Okay, blessings it. Okay, excellent. All right, so once again, my apologies. I have to, unfortunately, have to drop off children at school and then come back here for this class. So it is a little hectic. Anyway, so we'll try and do 8:15 from now on, barring anything too terrible. That should work out perfectly fine. All right, so we're continuing with genetics today. I think we were doing, or just about finishing up Mendelian genetics, and then about to move into non-Mendelian, right? So that is what we're going to do today. Now, I made a slight update to the Mendelian genetics PowerPoint presentation. So you're going to see a couple of slides here that are new, but I updated it on Canvas though, so you can always download the newer version whenever you have the time. All right, you should be seeing my screen. Well, I can turn up my mic real quick. All right, I don't know if that made a difference, but let's continue. Let's see. We're already Punnett square ready, so we're going to waste time on that. I won't bother too much with the revision today. I'll just jump right in. Spoke about cystic fibrosis already and Gaucher's disease, right? And you already know how to do a dihybrid cross. And we spoke about the basics of meiosis. Though do remember that we will be doing meiosis in some detail in a little while, right? And we did test cross 2. And I think we're somewhere around here, right? We're talking about incomplete dominance and codominance, if I remember correctly, right? As I said, don't have a lot of time to be running through them again, unfortunately. So let's move from there. All right. Oh, we got a good flow here, man. All right. I think we had gotten to as far as multiple genes, right? Well, that's where I'm going to start today. If I haven't reached here, you can let me know, but I think it's somewhere around here because we did codominance and incomplete dominance already, right? Now, in multiple alleles, the idea here is pretty simple. When you're doing Mendelian genetics, each allele had only 2 forms, okay? You had a dominant form and you had a recessive form, okay? in multiple alleles, you have more than two forms, okay? Now, blood is a good example. Blood type is a good example of multiple alleles because in blood, you can have allele A, allele B, and that there is allele for blood type O, right? Now, you notice the way I wrote A and B, right? So I is for immunoglobulin, right? Which of course is an antibody. And what this is saying is that your blood type can be type A or type B, right? Depending on the antigen you find on its surface, okay? The antigen is determined by, of course, this gene here. So if your blood type is like this, and of course, remember, everybody gets two genes, your blood type is A, okay? Now, if your blood type is like this, your blood type is still A. Why? Because, as you can see, this is written as a common letter I, which means blood type. Remember, this equals to blood type O. So written as a common letter, it means it's recessive, right? So one dominant gene, one recessive gene, you're still heterozygous for the dominant, right? And of course, on the other hand, we can have B like this. Or, of course, like this, right? And both of those would be blood type B. But because A and B are codominant, somebody with a blood type like this would have blood type AB. And the only way to get blood type O is to have two recessive genes, okay? So from those three genes, we have 1, 2, 3, 4, 5, 6 genotypes for blood, right? And 1, 2, 3, 4 phenotypes. So already we're getting more variants than we would in standard Mendelian genetics, right? All right. So another example here, as it says, is rabbit coat color is determined by a single gene with 4 alleles. You have the white wild type, which is sort of a reddish brown. Of course, it's written as a capital. You have chinchilla, which is sort of a mottled color. You have a Himalayan, which you can see has black patches and white patches. And you have albino. Now, you'll notice here that the wild type is written as just capital letter C, whereas chinchilla Himalaya, of course, are written out with a superscript, right? And the albino is just a common C. I mean, realistically, I should have written this as capital, instead of saying wild type, just a capital R, for example, for red, but you get the basic idea, right? So here we have our wild type, that's the red one. That's the chinchilla, right? Now, once again, it also mentions blood here, and we already went through blood. So remember, multiple alleles, you have more than 2 alleles for a trait, and those alleles can be any combination of dominant, codominant, or recessive, right? They don't all have to be dominant, they don't all have to be codominant, right? Now this is just showing you here the red blood cells and the antigen on the surface of the red blood cells. This is what gives you the blood type. And remember, these antigens here are controlled, of course, by the genes. Okay. Now, we also have something called polygenic traits. So when you have a trait that is controlled by more than 2 genes, right? Now keep in mind, I said genes, not alleles. So in multiple alleles, of course, you can have more than 2 types of alleles. So as we said before, A, B, O, right? Or the 4 different ones for the rabbit. In polygenic traits, you actually have different genes that are going to be influencing the same trait. And those different genes, of course, can have their own alleles, so, right? So it mentions here stem length in some plants, eye colors in fruit flies are controlled by 3 different genes, and human skin color is controlled by 4 different genes. Okay? Now, human skin color can get a little complicated. Now, because once again, you have multiple individual genes involved, and each one of these genes can potentially be dominant or recessive, you can get lots of different combinations of traits based on how the genes are inherited. So it gives you an example here of stem length. And it says, suppose stem length in a plant is controlled by 3 different genes, A, B, C. Each diploid plant has 2 alleles for each gene, right? So it gives you here an example, right? So we have, well, let's do our own example. Let's do one that is completely heterozygous. So since we have 3 genes, let's say we have capital A, which means the gene for tall. We have common A, which normally means short. We have capital B, which adds to the tall, common B for the short, capital C, common C, you know, the whole thing. So this would equal a tall plant, right? Now, if we had instead something like this, that of course would give us a short plant. I mean, that's kind of obvious, right? But what if we had a situation like this? Now, this plant clearly has genes for tall, but it also has in slot B, they're all genes for short, which means this plant is going to be somewhat intermediate. Right? It's not going to be super tall. It's not going to be particularly short either. Right? And of course, if we had a situation like this, and still had 2 genes for that, it will be, and this is not really a word, but let's call it shortish, or somewhat short, or moderately short, however you want to put it, right? So depending on how many of the dominant genes you get versus recessive genes, you can find the trait, which of course is height, being enhanced, okay? And of course, the more genes that are involved in this, the greater the impact will be on the height of the plant, right? Which is basically what we're explaining here. All right. Now, because of this idea of having multiple genes involved, you know what? I think I want to show you the human one real quick. Let's do human skin color real quick. It's one of those fascinating ones. All right. So human skin color. Now, you can see here that with this chart here, we're looking at 3 genes of human skin color, right? Just like we're looking at 3 genes for, what was it? The length of the stem, right? So even though, as it says here, human height is controlled by more than 4 genes. So we know, but there are lots of them, but obviously the more genes we add to it, the more complicated the whole thing will be, right? So in this case here, we're only examining 3 genes. And of course, you can see them named A, B, C, just like we did for the plants. So this is looking at the skin color distribution throughout a human population, right? Now, what you'll notice here, of course, is that this is a bell curve. Y'all should have done bell curve already, right? And a bell curve, of course, is what we call a normal distribution. Okay. Now, what makes this a normal distribution is this. When you are surveying a natural population that is uninterfered with by any other factors, you're expected to get this sort of setup for any trait that you're investigating. You know, except for traits, of course, that are in 2 forms only. So you can't expect a bell curve if you're checking the ratio of males versus females, for example, right? But whether it's height or weight or skin color, right? The number of offspring each mother has, this is the sort of distribution you expect to get in a normal population, okay? So a few people will have the extreme negative end of the curve, right? And a few people will have the extreme positive end of the curve, with most people being somewhat intermediate. Therefore, they sit in the middle, which is the highest part of the curve, right? So all of this should look familiar to you, right? Now, that said, let's start here. Remember now that each person is going to inherit, of course, 6 of these genes for skin color. 3 from a mother, 3 from a father, right? So this first person here, AB, which has capital ABC, for both parents, you can see they come out as the darkest possible skin color, okay? But as you start to change one of the dominant genes to a recessive gene, as you can see in both parents here, then the next person's skin color starts to lighten a bit, right? And as you add the recessive genes to the combination, of course, the skin gets lighter and lighter and lighter. until you get all the way down to your extreme recessive side, which of course you get only recessive genes. And the 0, of course, here is for the lightest possible skin color. So that's kind of obvious, right? Now, what this chart does hopefully suggest to you, I'm hoping you can see a couple of things that are impossible here, right? So let's see if this makes sense to you. Per 2 people, well, let me ask you, if we have 2 parents like this, can these 2 parents give birth to a child that has a skin color of 1? So here are my combinations. Can they give birth to a child with a skin color of 1? So this leads, of course, to this. Right? So what skin color is this person? Let me rephrase and say that. What skin color is that person based on the graph in front of you there? Let me see, who should I ask? Tasanya, what skin color is this person here?
Number 3.
Number 3, is it? Let's see. So we have one parent that is all recessive, one parent that is all dominant. So number 3 makes perfect sense. Thank you very much. All right. Now, what if instead of giving you genes, let's give you parents. Now, let us say you have one parent that is like this. Another parent just like this. Can these parents have a child with a skin color of 0? Can these 2 produce a 0 offspring?
Yes.
All right, I hear a hesitant yes from Blessing. Jodian, what do you think? Jodian Edwards, are you there?
Sir, could you repeat the question, please?
What the question I'm asking is, can these 2 parents have a child with a skin color of 0?
Yeah, yes, sir.
All right. Something is in the chat. All right. Yes. All right. One more person, and then we talk about it. Daniel, what do you think?
I think it's a low chance, but yes.
All right. So everybody's on the same page, and I actually agree. So what this is saying to you, though, right, is Jody, your mic is still open. You can
Oh, sorry.
no problem. So remember now, if you look at parent 1, what number you think parent 1's skin color would represent?
6.
All right. I agree with you. So parent 1 is a 6. And parent 2 is a what?
3.
3? Okay. Let's see. We have 1 fully recessive gene, and we have 2 heterozygous dominant genes, right? So we have 2 dominant phenotypes and one recessive, right? So what do we get for that? Well, this one here, so yes. So that gives us about a 3. So what we have here is a very, very dark-skinned person and a moderately dark-skinned person. And they can give birth to a child that is very, very light-skinned, right? So as the point I'm trying to make here, I suppose, is that sometimes you will see 2 parents and then you'll see their offspring. You're like, no, man, there's no way that child could belong to those people because the skin color looks so different from what you'd expect. Normally, you expect the skin color of the offspring to be somewhat in line with whatever the parents have, right? But this here is just showing you genetic proof that you can have 2 black people that give birth to a child with white skin. Okay? As long as they're not very, very dark, it's possible. As remember, they're very, very dark, it means they have overwhelmingly dominant genes, so it wouldn't work. If they're somewhat moderate or intermediate in skin color, it is definitely possible, right? So yeah, sometimes you can say black people would like picnic and kidnap them. That's not the case, right? Now, this is just giving you an idea as to what the numbers actually meant. So very light, of course, we're talking, it's the word Caucasian is very funny, right? So Ruth Ann, go ahead.
Yes, sir. How? Okay, I can understand if the example before could lead to like a 3, but how could it lead to the lightest possibility?
Okay. All right, tell you what, I have an empty slide. Let us say we have, you want me to use the same example?
Yes, that's fine.
All right, so we have, something like that, right? So what we'd have to do is we'd have to see the possible gametes that you can produce here, right? So which means we need all possible combinations of A, B, and C, which is going to take forever, but let's do some of them. I won't do the whole Punnett square, but I'll do enough to give you an idea as to what we're doing. Ugh. All right. Did that one already. You can see how complicated it gets with even 3, right? But it's good practice, right? So this is the one that I'm interested in anyway, right? Because this gamete has all recessive traits. Over here, of course, we can do the same thing, but to speed things up, I'm just going to show you that we can still get a gamete like this, because we have small a, small b, small c here, with all recessive traits, which of course means that both of these, if a parent produces each gamete with all the recessive traits they have, they combine into the child, and the child comes out like this, which means the child is fully recessive, which means that they're the lightest skin color possible. So even though one parent is very dark, the other parent is moderately dark, there's a slim chance, don't get me wrong, it's slim, it's about 1 in 64. I mean, how many people have 64 children, right? but there's a 1 in 64 chance that they could produce a child that is as light as is possible to get, right? Because they do have the genes in them. It's just the probability of those genes being put into the right gametes and those right gametes meeting that makes the difference. Does that make sense?
Yes, sir. Thank you.
All right, no problem. All right, so this is showing you the skin colors from 0 to 6. Honestly, it is very dark. It's not even that dark. I think I mean I've seen some people black enough you can write on them with a white marker and it look good. Anyway, now I think I was saying to you that very light. The word Caucasian is funny. Do you know white people, quote unquote white people, you may know this, were only supposed to be people from the Caucasus region. I think that's somewhere in Europe, right? Nobody else was white. Irish weren't white. Italians weren't white, right? Certainly, people from North Africa weren't considered white for the longest time. But in the last 80 to 100 years, we've bundled them all together and called them white people, right? So even you want to understand this, the concept of skin color in humans is and how it's represented is somewhat fluid, right? Because somebody who's very, very light like this is the kind of person that when the light shine on them, they don't tan, they burn, right? While this here is closer to, I guess, what you consider to be your standard white person, which means that they do tan. But this person would not have been considered white up until the last 100 years. If that makes sense to you, right? And oh, by the way, also, there are very light-skinned black people in Africa, which seems to defeat the purpose of calling them black. But once again, you have to understand the recessive genes for skin color does appear in the black population, right? It just appears at a much lower rate than in the quote-unquote white population. And here's the reason why. And see if this makes sense to you. When this is around 75,000 years ago, 70,000 years, we had groups of Africans leaving Africa, going up to North Africa, and then from there, spreading into Europe, right? Now, these people were black people, right? And as they moved into Europe and went further and further north, a couple of things happened. One is that the environment changed dramatically, right? It was a lot colder, for one, right? Snowfall and all kind of things. Food availability was not what it was before. And active sunlight was much less, particularly over winters, okay? Now, in Africa, the last thing you have is a lack of sunlight. So in Africa, of course, I'm assuming everybody knows that UV radiation damages DNA, right? So melanin evolved in black people to preserve DNA, okay? But you also need some UV radiation because you need to turn a precursor to vitamin D. I think it's called procalciferol. Don't quote me, but I think it's that. To calciferol. And then this is turned into vitamin D at the liver, okay? So you need UV for this, right? When you're in Africa, you have more UV than you know what to do with. So you need melanin to actually protect the DNA in the cells to stop from getting skin cancer and killing you. When those peoples left Africa and started moving their way, keep in mind it took thousands of years, right? Up north, they were to get less and less sunlight. So all of a sudden now, that very dark skin would have reduced their access to the UV radiation they would need to make that calciferol. And without vitamin D, of course, you get rickets. Right? Rickets, very soft bones, right? Deformation in the skeleton, which means you cannot function properly. You're a burden to your tribe. You die, you don't reproduce. Occasionally, a child would be born with one of the mutated genes for lighter skin color, right? Because they would have been coming from Africa. These genes would have been in the African population, right? But because the environment would favor people with more and more of these recessive genes and pretty much slowly kill off the offspring that have too many dominant genes over time, you'll find the skin color of the population getting lighter and lighter and lighter, right? So this change in skin color is evidence, well, of evolution. Because we know that the human population began in Africa. So all humans are genetically Africans. But difference in environmental conditions would have been enough to bring forth changes like the change in skin color, right? Even the hair on the head, why people who live in temperate conditions tend to have straighter hair. It's because straight hair holds heat better. So it insulates your scalp, and your brain, right? I don't know if any of you all have straight hair versus curly hair, but I'm sure you know that when you have your hair plaited out, cane or whatever you want to call it, it is much easier to deal with the heat than when you have your hair cream and straight. Creamy hair hot out hell, because that is what it's for, to insulate the skull, right? Even the shape of the nose of a Caucasian person and the amount of hair in the nose. Caucasians tend to have hairier noses than Africans. Why? Because they have to filter snow out of the air as they breathe, right? Breathing in dust is bad, but breathing in snow can kill you, right? So yeah, if you ever wondered why some populations have a higher concentration of lighter skinned people than others, as in Caucasians, it is of course genetic, right? Now, the extremely light skin color, of course, would favor people who are closest to areas that have very, very long winters. If you have a situation in which you have winter and it gets a little overcast and it's cold, but it's not detrimentally cold, like in the Mediterranean, you will get a lighter skin color, but nowhere near as light as that. You will tan easily because, of course, you will have days which have extreme sun as well. So you can actually find Mediterranean, quote unquote, Caucasian or white people that are darker than the quote unquote black people, right? All right. Hopefully that makes sense, right? Now, let's move on. This here is just showing the same thing we spoke about with the skin color. And as I said too, if you have a 1 in 64 chance here of these two people producing a child that is either very dark or very light. So this is saying two people with intermediate skin color have a 1 in 64 chance of producing a very, very dark child or a very, very light child, right? I've seen pictures of like sisters that are like this. One is an extreme in, you'd think she's white, and the other one is almost is extremely black. It does happen. But obviously 1 in 64 is pretty rare, right? All right. Probably I don't know if you're saying something I can't hear. All right. Now, environmental influences, we just spoke about some factors, right? As it says, an organism develops, many factors can influence how the gene is expressed, or even whether the gene is expressed at all. Influences can be external or internal. All right. Let's take height. I think it's something I've mentioned before, right? But their height is also multiple allele, polygenic, right? Multiple genes determine how tall a person is. Now, you obviously can have a situation in which you have two people of moderate height for their sex. So you can have a man who is 5'9" and a woman who is 5'5", and they can have a child that's 6'3", simply because the child will take more of the genes for height, the dominant ones, than it would have been in both parents, right? Just the same way with the skin color. Now here's something you may not know. Generally speaking, right, there's a minimum height to the male child, but not to the female child. Generally, so keep in mind, this is generally speaking. This is assuming the child has good health, otherwise is well fed and everything. Generally speaking, the male child will not be shorter than his mother. So if the mother is 5'5", the child is not going to dip below that. The mother is 5'9", the child is not going to dip below that, right? The male child. All right. Anyway, let's link it back to the environment. Let us say you have a child that has gotten now genes that should make them 6'4" tall. They got several good genes for height. There's a famine. All that is available is a limited amount of carbohydrate-rich food. And for years, they're subsisting on this carbohydrate-rich food with not a lot of fat, not a lot of protein in there. In order for you to get to height, you need protein and fats for muscle. You need minerals that build up your bones. If these things are not in the diet, even though the genes are there to be 6'4", that child ends up being 5'5". That's an environmental factor that influences the expression of the gene. The genes want the boy to be 5'5". There's just not enough resources to make him 5'5". Therefore, sorry, to make him 6'4". Instead, all he can get is 5'5". But the 6'4" genes are still in him. So if things get better when he's an adult, he then finds a lady, and well, I mean, it's pretty much gonna have to be a lady, right? And have children. He's in a better position than his parents were. That child is now properly fed. He has passed on his genes for height to that child. Now the environment is different. So even though the father wasn't going to be 6'4", the son now will. They have the same genes, but the environment is different. Therefore, the son will grow nice, big, and tall, even though the father could not have, even though they have the same genes. Okay? So genes do not exist in a vacuum. You won't get something without the genes, but if you don't have the proper environment for the genes to function, you also won't get it, right? It's like IQ. And a lot of people don't like to hear this, but it is the truth. Your intelligence is set at birth. There's very, very little that you can do throughout the course of your life to improve your intelligence, right? One of the things that we've noticed that helps with intelligence in the first 2 years is this. breastfeeding the child. Breast milk has been tailored by nature for optimal development of the child. No matter how good formula is, it's not as good as breast milk is. And on average, breastfed babies are 5% smarter. You might not think 5% is a whole lot, but it makes a big difference, right? But remember, this is just for the first 2 years. Now, if you're asking, hey, so studying and doing all sorts of reading techniques and whatever, won't that make you smarter? No. That just gives you more information. And giving you more information, of course, helps with your ability to use your intelligence. So going to school does not make you smarter. You're born with your IQ intact, and it's not going to change very much. It is the additional information you get from going to school that allows you to use your native intelligence in better ways that makes you appear more smarter when you go to school, right? There's a hand up. Ruthann, go ahead.
Yes, I just wanted to ask what is classified as smart?
In terms of IQ numbers or in terms of socially, what are we talking about here?
Well, in terms of like how you were using it in terms
All right,
of
let's think of it in terms of IQ, intelligence quotient, right? Generally speaking, the IQ is adjusted to 100 for the population, right? So in any given country, your average IQ is going to be 100. That would be considered normal, okay? Now, keeping in mind that this adjustment for 100 is realistically the norm of what you'd get in like the United States or England or one of these Western countries, right? Because they're the ones who came up with this thing, okay? Now, in order for you to be considered a smart person, we're not talking about a genius, just smart. An IQ of somewhere about 120 would be expected, okay? So IQ of about 120, these are the kind of people that will get like a master's in business or something like that, right? So they're reasonably good at utilizing information, coming to conclusions, right? That said, a very intelligent person, somebody with a master's or a PhD in a science field, on the other hand, needs an IQ of over 130, right? Normally in the mid 130s, which is an interesting contradiction, right? Well, not really a contradiction. So you have social science smart and you have science smart. Science smart is harder, right? There's a reason why you have so many people with masters in psychology and all kind of nonsense, because it's once you have an IQ above 130, it's easy. It's hardly any work at all, right? When you're doing sciences, you need extra intelligence, right? I don't know if I answered the question. I think I may have gone off in the weeds.
Thank you, yes.
Okay, but generally speaking, if you have an IQ of 120 or above, you're considered to be fairly intelligent or smart, let's call it, right? That said, even though you're born with a set IQ, your environment can diminish your IQ, which is kind of sad, because your environment won't improve it very much, but it can diminish it massively. So for example, if once again you have a child that during their developmental years, their diet is very restrictive, very low in proteins and fats, it can shave off 15 to 20 IQ points easily. So you can have a child that should have grown up to be of average intelligence 100, and they are now mentally considered to be disabled at 80, right? simply because that they're not properly fed as they're growing up, right? Giving them extra good food won't make them smarter, but not giving them enough good food makes them dumber. Which is why, and keep in mind, once this pattern is set, it can't be unset. Once you lose those 20 IQ points, you can't get them back. There's a reason why so many countries spend a lot of time on thinking about nutrition, children's nutrition, because you can permanently damage that child who will become a member of your society and a less functional member of your society, okay? So external, so that's what I mean when I say environmental factors. Oh, let me mention one more thing too. It's a little depressing, but you need to know. If you make it into the med program, you can assume your IQ is somewhere around 125 or higher, okay? Just by virtue of you make it into the medical program. The by virtue of you make it into pre-med, your IQ is at least 115, right? The average IQ in Jamaica, when adjusted for comparison's sake to the average IQ in the US, is 70. Which is, I always find it so depressing. In the United States, an IQ of below 85 is considered too mentally incompetent to join the army, to run around with sacks on the back and follow basic instructions, right? The average IQ in Jamaica is 15 points lower than that. What this means is that we have on average a population whose IQ is not conducive to the ability to follow simple instructions efficiently, right? If you have spent any time around your average Jamaican, you will start to get frustrated because some of the things you find very obvious, they just don't get it, right? So keep that in mind. You all in particular, more likely than not, you've spent the majority of your lives around people with an IQ of 100 or above. You have no idea probably what the average Jamaican is like intellectually, right? So when you're trying to talk to an average Jamaican and it's like you're talking to a brick wall, please understand that literally they might not be able to understand you. Anyway, it's just one of these things you have to get used to. And it's not about you feeling superior to anybody. It's about you understanding that everybody can get what you get. You understand what I'm saying? Okay, moving on. And by the way, Jamaica does not have the world's lowest IQ. Okay, no. The lowest IQ is oh God, I can't remember. It's an African country. I think it's Democratic Republic of Congo. They're somewhere in the 60s, low 60s for their IQ. Right? Anyway. External influences, we mentioned food. Obviously, from a human perspective, food is going to be important. Plants, a little bit more complicated. Temperature, of course. And of course, nutrition like everybody else. Temperature. Do you know that olive trees can grow in Jamaica? I don't know if any of you have ever seen olives growing in Jamaica. We actually have people who have olive trees as ornamental plants in Jamaica. Those olive trees will never produce an olive in Jamaica, right? Or the quote-unquote American apple trees. You can grow American apple trees up on near Blue Mountain. I don't know if you know this. You can plant them up there. You can grow peach trees and pear trees. All of these things can grow up there. So why don't we have these things in the local market? It's because even though the temperatures up in Blue Mountain are cool enough for these plants to grow, these plants require a period of frost before they can flower. So their roots have to freeze up. So the ground has to get frozen. And that tells the plant that spring is coming, it's time to put out flowers. So the pear trees and the peach trees and all of these other ones, they're never going to get that frost because it doesn't frost over in the Blue Mountains. So even though they can grow, they will never produce fruit. Same thing with the olives. Olives are a Mediterranean plant, and the Mediterranean winters, you do get days of frost on the ground. So it gets cold enough ice to freeze up the soil. It doesn't last very long, but it happens. When that happens, the olives know, yes, it's time to flower. So we'll have olive trees in Jamaica, but you will never get the olive off of it. I mean, I suppose if you go out there with some liquid nitrogen, I guess, and degrow the plants, or if you plant in a big pot and then roll the pot into a giant freezer or something, that would work, but it's definitely not viable, right? So that's it. Those are hopefully you can say those are examples of how environmental influences can affect the way phenotypes are expressed. In this case, the phenotype we're expressing is sexual maturity, okay, in terms of the plants, right? All right. Now it also mentions internal influences. So we have here internal environments of males and females are different. Of course, it's going to be because the hormones and the structures are different. I want you to understand that there's no such thing as a male hormone or a female hormone. We simply have hormones, right? The structures in males and females, some of them are a little different. And because of that, the hormones have different effects, right? But males have estrogen, females have estrogen, males have testosterone, females also have testosterone, right? And the difference, of course, is going to be in the volumes of these hormones that are being produced, as well as how sensitive we are to these hormones. So males are more sensitive to testosterone than females are. There was a question, I missed it. Okay. So it mentions here male pattern baldness in humans. Male pattern baldness is caused by sensitivity to testosterone. Right? So some males are hypersensitive to testosterone. And this testosterone builds up on the scalp, particularly in the middle of the head, right here. As the levels of testosterone build up here, that's something to do with exposure to the sun, I think. Right? Or is it standing up? I have to check it out. So testosterone builds up here first, right? And that extreme level of testosterone kills the follicles of the hair and causes the hair to fall out. Now, females never have that much testosterone in them, and females are not nearly as sensitive to the effects of testosterone as males are. So females will not get pattern baldness like this, right? It also mentions the feather colors of peacocks. So the peahen over here is very drab and, if I'm honest, kind of ugly looking. Look at her feathers, how pathetic they are. These pathetic feathers are very good for blending into the environment and keeping them safe. Look at how vibrant this guy is, right? Same hormones, but the males are more sensitive and their feathers respond differently to them. These feathers are there to impress the females. This also makes them stick out like a stick out like a thumb, which means the life expectancy of male peacocks in the wild is much lower than that of female peacocks, because they tend to get eaten because it's kind of hard to hide when you look like a gay pride parade, right? But this increases the number of offspring they have. So now you have to trade off. Do you want to be drab like the females and then get no love? Or do you want to be blinged up, not live as long, but have plenty of kids? And this is nature's answer. It's better to be blingy with lots of picnic than to be drab and alive, right? That's what nature says, not me. Okay. It also mentions age. And this one is a little difficult. As people get older, certain genes don't seem to express as much as they would in a younger person, right? For example, we know that genes that control the production of lymphocytes, which are white blood cells, as you know, don't seem to work as well as you get older. And because of that, older people are more susceptible to viral diseases, bacterial diseases, right? Once again, it's a complex process. Human beings are still working on it. But we know the older a person gets above, let's say, the age of 40, 50, the more susceptible they are to disease and death, right? I mean, fundamentally, it's because nature is only interested in your ability to reproduce. Once you've passed the age of proper reproduction, nature's happy to kill you off, right? Doesn't care. Now, all right, so let's move on now to talk about sex determination. This is, of course, nice and easy in humans. Everybody knows humans have 46 chromosomes, and they are set in 23 pairs, okay? 22 of them are called autosomes, and these are your regular chromosomes. And then you have one last pair, which are your sex chromosomes. Now, generally speaking, females are XX and males are XY. I'm hoping you notice that I said generally speaking. There are medical conditions that will make things a bit ambiguous, and we will talk about those when we talk about mutation, right? Now, when you look here at the X chromosome and the Y chromosome, I'm hoping you notice that there's a stark difference. The Y chromosome is the smallest human chromosome. It has the fewest genes. So it's small, there's not very much in there. But every single one of those genes is a dominant gene, right? So even though there's not a lot of them, they make a dramatic difference. They cause changes to multiple organ systems in the body, right? So those few genes have a very wide-ranging effect on the body, even though there are not very many of them. Now, this little Punnett square here is showing you the probability of having a male versus a female, right? Offspring. So you know XX, of course, is from the female. All she has are X chromosomes. Males, of course, have X and Y. Now, hopefully, you realize from this that it is not the female that determines the sex of the child, it's the male, okay? All the female can produce are X chromosomes. Therefore, it is the father that makes the child female or male, not the mother, right? Now, if we look at this Punnett square here, you get the impression that you have a 50-50 ratio when it comes to offspring, okay? So whenever there's a pregnancy, there's a 50% chance that it's male and a 50% chance that it is female. That seems kind of obvious, right? Here's the thing. There is a slight bias towards female offspring, right? Being born. So slightly more female offspring will be born than male offspring because of a couple of factors. One of them is the XY setup. Many very bad genes can be found on the X chromosome. What we can refer to as lethal alleles. All right? And these lethal alleles can, well, I shouldn't say many, but there are a few. These lethal alleles are usually recessive. If you are homozygous recessive for these lethal alleles, the offspring tends to die before childbirth. Okay? Now, if you're a female, you are usually going to have two X chromosomes. Let's say one is normal, like that. Or it can be like this. So you have normal and you have one lethal allele, which still means that you're perfectly fine, because the lethal one is, well, I wrote it as a superscript, but that should really be recessive. The only way for a female to die from a lethal allele is to have two copies, right? As long as a male has one copy of the lethal allele, he's dead, because he only has one X chromosome, right? So therefore, males are more susceptible to genetic disorders that can result in a termination of the pregnancies than females are. So there's a slight, so statistically, you would think that is 50-50, and the Punnett square which is 50-50, but there's a slight bias towards females, okay? Now, to further that idea about some chromosomes being on the X chromosome versus Y chromosome, we're going to talk now about sex-linked inheritance, all right? I just said to you that you can have bad chromosomes on the X chromosome that can lead to the termination of the male fetus. But you can also have bad chromosomes on the Y chromosome as well, right? So whenever you have a trait, or the trait, of course, is going to be controlled by the alleles, right? So the traits are going to be sex-linked if they are found, of course, on your X or Y chromosomes, right? As I mentioned, there are traits controlled by genes located on the sex chromosomes. That's all that is. That is, the alleles are written up as superscripts of X and Y. That's what I was doing before here. And the Y-linked traits are passed only from male to female. I mean, that's kind of obvious. You can't be a female with a Y chromosome. Okay? Since males only have one X chromosome, there's a gene on the X chromosome. Males get one copy, like we said before. And of course, that one copy must have its effect. Okay? Now, we have an example here of fruit flies. Fruit flies are one of these insects that have been heavily used in genetics, right? So it mentions here now the gene for eye color is on the X chromosome. You have a red eye gene and a white eye gene. You'll notice they've written recessive here. So you have X superscript R and being used here, right? This is to show what sex chromosome is on, of course. And then, of course, we use dominant and recessive here, so we can identify which one is recessive. And it mentions here homozygous red-eyed females, white-eyed males. What do you get? So here we have it. We have a female that is homozygous for red eyes. So she can only produce genes for red eyes. So there are the two genes and the two gametes for her. The male, of course, who has white eyes, can only produce one X with the white eye gene and a Y. That has no impact at all on eye color, right? So of course, if we fill in the Punnett square, you end up with this. So we have two females that have red eyes and two males that also have red eyes. So everybody here has red eyes. But note, the two females are heterozygous for red eyes. which means that there's a chance that they will have males, their offspring, that are going to have white eyes like their father. All right? So this is showing you a case in which a trait can skip a generation. Okay? So the father has white eyes, all the children have red eyes, like their mother, and then some of the grandchildren pop up, but the males will come back with white eyes like their grandfather, right? So the trait is skipping a generation. All right, so this is all that this is showing that. So heterozygous red-eyed female, red-eyed male, and it shows, of course, that when you cross now the females who are heterozygous with the males that are red-eyed, you can end up, of course, with some males, once again, that get back the white eye trait that their grandfather would have had. Okay. Now, let's talk about pleiotropism. This is the last one of these before we move on. Okay. Pleiotropy. See if this makes sense to you. When a single pair of genes controls the production of many characteristics, then it is called pleiotropism. And the gene is a pleiotropic gene. The term was coined in 19, nobody cares. So one gene impacting multiple traits equals pleiotropy, right? So the basic idea here is that it describes the genetic effect of a single gene on multiple phenotypic traits. Mutation in a pleiotropic gene will therefore have multiple impacts. So if anything happens to that one gene, you're not simply going to get one trait change, you're going to get multiples of them, right? And the mechanism of pleiotropy in most cases is the effect of a gene on metabolism pathways that contribute to different phenotypes. It's basically saying that chances are the pleiotropic gene is impacting some enzyme or the other, right? That is used in different processes in the body. And once you affect that enzyme, you're going to affect the way the products are formed, right? All right. So let's give you some examples now. It mentions here that in Dubzansky in 1927 demonstrated that a gene for white eyes in Drosophila may affect the shape of the sperm storage organ in females, as well as other structures. So the gene for eye color affects the reproductive structures as well. Pleiotropy. Another one here, PKU disease. This is phenylketonuria, is an example of pleiotropy in humans. So PKU is due to a mutation in a single gene, the PP gene, that codes for the enzyme phenylalanine hydroxylase. So phenylalanine hydroxylase converts the amino acid phenylalanine to tyrosine, right? Now, when the mutation occurs, you pretty much do not get the tyrosine because the gene is damaged and the enzyme is damaged. And it can cause mental retardation, reduction in hair, and skin pigmentation as well, okay? So the PKU gene codes for an enzyme which converts phenylalanine to tyrosine. So one amino acid is turned into another amino acid. You need tyrosine, right? Without the tyrosine, the skin and hair have reduced function, and it affects the brain as well. So 3 impacts from one gene. And once again, as you can see here, it's an enzyme. You mess up the enzyme, and the enzyme's product has multiple impacts. And of course, you don't get those impacts, right? All right, one last example. Sickle cell anemia. This is one that you're more likely to be familiar with, right? In sickle cell, as you know, this is also one gene different. You have sickler hemoglobin, and you have normal hemoglobin. The difference between these two is one gene, right? In the abnormal hemoglobin, it crystallizes when it gets exposed to oxygen. Sorry, when oxygen. Okay, let's start again. When somebody with sickle cell anemia makes their red blood cells, okay, they start out looking perfectly normal. So these are normal red blood cells over here, right? When you expose these normal looking red blood cells to low oxygen conditions, which is inevitable. I mean, they deliver oxygen. At some point, they're going to be empty, right? It causes the hemoglobin molecule to change shape, to become crystallized. And when enough of these hemoglobin molecules become distorted, they distort the cell they're sitting in. So the cell itself starts to change shape. You end up, of course, getting these quote-unquote sickle-shaped cells, right? Now, you could say, okay, that is one effect. But what else does the sickle cell gene affect? It increases risk of heart failure. It causes the sickle cell cells to form clumps and clogs in small blood vessels. They accumulate in the spleen. So they damage the spleen. They can damage the joints in the body. Of course, you have the anemia. They can cause brain damage, paralysis, rheumatism, kidney failure, one whole cascading host of negative impacts, all because of one gene. Okay? So, somewhat pleiotropy. Okay? So you have multiple negative impacts caused by one gene not functioning properly. Okay. Now, one last thing I want to mention about this thing. When human beings were cataloging the genes that make up the human, this was the Human Genome Project, right? So Human Genome Project. So scientists all over the world pretty much try to find every single human gene possible and catalog them. Now, when the project began, we assumed human beings had somewhere around 66,000 genes. Why? Because chimpanzees have that. Okay? So chimpanzees have about 66,000 genes. Human beings are very genetically related to chimpanzees. So we figure, hey, if they have 66,000, we're supposed to have somewhere near there as well, right? But when we're all done cataloging, we realize that human beings actually only had around 30,000 genes. Right? Which means we have lots of genes in us that affect multiple structures in our body. Right? So mutations of any one of these genes with multiple impacts can give rise to a whole host of genetic problems. And when we do mutation, we'll talk more about this, right? So this is what happens when you have one gene doing multiple duty. When it goes bad, you can have wide-ranging negative effects, right? Okay, that said, let's talk about epistasis. Now, in epistasis, you have genes impacting the way other genes function, right? So one gene influences the way another gene is expressed. is another way of putting it, right? So you have two genes, one of them makes a protein, but it can only make the protein if another gene tells it to make the protein. So one gene impacts the way another one is expressed, right? It mentions that these genes can be on separate alleles and segregate independently. So even though that gene is impacting the other gene, they don't even have to be on the same chromosome, right? It mentions here in the case of a Labrador retriever, I'm sure you all know what a Labrador looks like, right? I think they're one of the most popular dogs in North America. I've even seen them in Jamaica. By the way, certain dogs you should not have in Jamaica, right? So Huskies and those kind of dogs should not be in Jamaica. Labrador really should not be in Jamaica either, because they have a very fine coat and the heat is very detrimental to them. It's wicked to carry them into the island. Anyway, so the Labrador retrievers, who should never come to Jamaica. Let me get rid of some of this. Have a gene E here, which controls the expression of another gene B. Okay? So gene E controls the expression of gene B. Now, let's talk about dependent gene epistasis, right? As an example of epistasis. So this is what we're talking about now. In mice, it says here, one gene controls the production of the coat pigment, and another gene controls the dilution of the pigment in the hair, with dense pigment being dominant over dilute pigment. So this is very much like Siamese cats. Okay, we don't want that example. But one important difference is that pigment gene is epistatic over the dilute gene, because the recessive allele of the pigment gene is a mutation that produces no pigment at all, so there's nothing for dilution to work on. Okay, let us use this now, and we're going to do a little Punnett square to see if we can make it make sense, right? All right, so according to this table here, we have two genes. We have gene B and we have gene D. Sorry. All right, so now what we're looking for now is B here is going to control how much color is put in, right? And D here, wait, sorry, it's the other way around. So the black pigment, so that's the color. You have black pigment, you have no pigment. Another gene that controls the dilution of the pigment in the hairs, with dense pigment being D and dilute pigment being small d, right? And it says the pigment gene B is epistatic over the dilution gene. Okay, I got it. All right, so here's the basic idea. This gene gives you black, right? This gene gives you no black. This gene gives you intense black, right? So this one makes the black color intense. And this one here makes the black color light. So more like a grayish black than a dark, and not a very dark black, right? What they're saying though is that if you have a case like this, this animal will be black. Okay? Now, if you have another case where you have one that is all recessive, this one is going to be white. Right? Now, let's say you do your normal cross and you get, of course, F1 generation, like you'd get this. Or what am I saying? Let's say you do your parental cross. You get F1 generation like this. And then you cross two of these. So of course we have to do the gamete thing. Right? And of course the other parent has the same combination, right? Then we quickly do a Punnett square. Doing. And here all this goes. So let me just do it quickly. Okay. So here's my Punnett square. Now, change the color here. Now, ordinarily, when you look at this, wait, I'm not done. I didn't even... Okay. This, of course, looks like a dihybrid cross, which it is, right? Now, the difference is we're only going to get two possible outcomes. Either we get a black color or a white color, okay? So even though we have two genes, we only have two potential phenotypes, okay? So the first one, for example, here has two capital B's for black, two common D's for dense fur. So this guy is going to be black, okay? Next one here, two capital B's. You have a capital D there, so they also have a black fur. Same thing with this one here. Same thing with that one. This one is also going to have a black fur. Now, when we get here now, we have two capital B's for the black color, and we have two common D's, which means that the intensity is low. So this one here is going to be gray in color, okay? Well, the other one is black. There's another one that is gray. Here's another black one. And here's another black one. You see how common the black ones are. Now, here's where things start to get interesting. Because we have two common B's here and two capital D's. So the capital D's say make the color intense, right? But the two common B's say don't make any color at all. You can't make an intense no color, which of course means you default to white. Can't be gray, you have to be white. And the next one here, of course, is also going to be white. This fella down here is black. This one here, two small D's, but it has a capital B, so they're gray. This one here, two small B's, so they're automatically white. And the last one is also automatically white, okay? So if we now start to look at the phenotypic ratio to see what we get, we have what? One, two, three, four, 5, 6, 7, 8, 9 black ones, right? Then 1, 2, 3 gray ones. The second row, third column, should have a common B instead of capital. Okay. Second row, third column. Ah, yes, one of these B should be, this should be a common B right here. So you're absolutely right. So that should be a common B. Right? She's right. Right here, so. Okay. Thankfully, it doesn't affect the results. That said, if you're given a question that anything and made a mistake like that, you would lose marks for it. So she's absolutely right to point it out, okay? Okay, so now it's more accurate, good. Thankfully, it hasn't affected the ratio, but you would still, as I said, you would still have lost marks because it is an inaccuracy, right? So it still has to be corrected. Now we have 4 whites. Right? So this ratio looks like no other dihybrid ratio you've ever seen. Because this is 9 to 3 to 4. Okay? And remember, it is a dihybrid cross, but functionally, we're looking at one pair of genes on or off, right? And then they can be recessive or dominant, okay? So the first gene here, which is the black gene, determines color, no color. And the second gene, which is the D gene for intensity, equals very dark color, very light color. But they're all affecting the same color, right? All right, then we have something called enzyme pathway. In enzyme pathway here, what we have happening is this. We have multiple enzymes required for the production of a final product. This here, for example, is the color of a flower. To make the color purple, you want the flower to be purple. You start out with a white compound, which is compound A. Then we have a gene, which is gene P. And gene P makes the enzyme P, which converts compound A to compound B, which still happens to be white. Then now compound B is worked on by enzyme Q, which is produced by gene Q, to finally give you compound C, which is purple, okay? So you need to have these, in order to get your final product, you need two genes which make two separate enzymes, okay? Now, any sort of disruption in any of these genes is going to cause a problem. So for example, if gene Q stops working, you still have plenty of enzyme P, which means, of course, you get a buildup of compound B. So the plant flower still looks white because compound B is white. So you get a reduction in compound A since it's being converted to compound B, but since both of them are white, the flower stays white, right? If you have gene P getting damaged, then you have no enzyme P, so compound A is never converted to anything else, and it stays white. So the flower also remains white. So any negative impact anywhere at all in this pathway, you end up with a different compound from the final compound you want, right? Therefore, we have multiple genes affecting the outcome, right? Hence, it is a form of epistasis. Multiple genes affecting the outcome, right? So enzyme gene P affects the ability of gene Q to function because gene Q is making an enzyme, and without enzyme P giving us compound B, then all the enzyme that is being made by gene Q serves no purpose, right? So gene Q therefore serves no purpose. All right. This is the last one I'll do for today. Duplicate genes. Real simple concept. We have compound A again, which is white. And in this case here, we have compound B now, which is purple. So this is once again flower colors. Note that you have gene P making an enzyme P and gene Q making an enzyme Q, and both of them are converting compound A to compound B, which of course means if gene P stops working, you still have gene Q, which means you will still get the final product, which is compound B. You might get less of it, so the plant might be a lighter purple, but you're still going to get the final purple. The only way for you to stop this process is for both genes to stop functioning at the same time, right? Now, if you think about it, certain substances that your body produces, it makes sense to have a situation like this when you have multiple enzymes all assisting in the production of that one thing. So if anything goes wrong, a damaged gene or anything, the organism doesn't simply die. It has a backup gene that allows for this process to continue, right? Might not continue as well as it did before, but it still keeps on going, right? All right, 9:55, I'll stop here because this is going to take a little while. This is looking at another epistasis again, and I'm going to ask you what sort of epistasis this is. But we'll do that next week, Tuesday. And those are Labradors, by the way.
Sir,
Go ahead.
I wanted to ask a question. I wanted to ask you if you could go back to the stem length example. Yeah, wait, not that one. The next slide.
I think I
No.
cleared it. I think it
No, the one that has the answers on it.
answers this.
Yeah, the next one up. Yes, that one. Sir, how did you arrive at the 10 centimeters again?
Okay, the idea here is that each one of these genes adds to the length of the stem, right? Remember, this is we're looking at polygenic traits, right? So each dominant allele increases the height of the organism here. So when you have 3 dominant alleles, they all add additional height, which makes the plant taller, right? The recessive alleles add a little bit of height, but not nowhere near as much, right? So the tall genes add more height than the short genes adds, but they're all adding height, if that makes sense, right? So therefore, the more dominant alleles you have, the taller you're going to be. So the 10 centimeter one here, we have 1, 2, 3 dominant alleles, 3 recessive. In the 8 centimeter one, we only have 2 dominant alleles, which means it gets less height from the genes, from the alleles that are left over. And the third one here, which is 14, has 5 dominant alleles out of 6, which gives it even more height, if that makes sense.
Okay, sir.
So it's a cumulative effect. It's just that the dominant ones give more than the recessive ones, but they're all still contributing.
So, sir, how do you determine the number? Because I see it here that the uppercase contributes 2. So is it that we add
Keep in mind, this is an example, you know, which means the numbers are made up.
Oh,
So they're arbitrary. It's not like because hey, there's a gene that gives exactly 2 centimeters. It doesn't quite work like that. But the idea here though is still that the dominant genes add more to the height than the recessive ones. So the more recessive genes you have, the shorter you're going to be. That's the basic idea here, right? Which is very similar to what happens in terms of human height, right? All right, any other questions? All right, folks. So I think we have another class this afternoon, this evening. So I will see you all at 5. All right, folks, have a good day.